Hi, I'm Art Krieg. I'm the founder and chief scientific officer of Checkmate Pharmaceuticals, and I'll be telling you about Vidutolimod, a virus-like particle delivering a CpG-A DNA TLR9 agonist. Our forward-looking statements are on the next slide and can be accessed on our website and regulatory filings. And I'd like to start by acknowledging the fact that most innate immune activators have actually failed in human clinical development. And what I mean by failed is that although most agents have very strong anti-tumor activity in mice as monotherapy or in combinations, in humans the monotherapy efficacy has usually been less than 5% with limited value in checkpoint inhibitor combinations. And this includes a variety of different STING agonists, TLR7/8 small molecule agonists, either conjugated to lipid tails or antibodies, CpG-B TLR9 agonists that I was involved with back in the Pfizer days, and also others. And I'll just note these have a phosphorothioate backbone that we now know can't be cleaved during TLR9 activation, and that limits the interferon response to the CpG. Same thing with CpG-Cs that have phosphorothioate backbones, and recently Vidutolimod failed in clinical development with that. There's one agent, however, that really stands out as being differentiated in terms of its activity, and that's Vidutolimod, where in monotherapy, a clinical trial in PD-1 refractory melanoma, there was a 20% ORR, 8 out of 40 patients responding by RECIST, 22.5% by iRECIST. In a combination with Pembro in PD-1 refractory melanoma, slightly higher response rates, 23% and 28%, N of 98. And in combination with Nivo in PD-1 naive neoadjuvant melanoma, a 67% pathologic response rate and 90% relapse-free survival at 1 year. So what's different about Vidutolimod from all of these other agents? Vidutolimod is a virus-like particle delivering a CpG-A DNA. And the first point is how distinct this CpG-A is. CpG-A DNA is biologically unique in its structure. It has poly-G regions at the end and a native DNA backbone that can be cleaved to give strong interferon induction. And the CpG-A forms G quadruplex structures that mimic viruses and retroviral DNA structures. So this is a very clear danger signal to the immune system. The CpG-A activates plasmacytoid dendritic cells, and so I want to talk a little bit about the PDC. Now, in their unactivated state, a very nice study from a French group showed that PDCs are negative for PD-L1 and CD80. And when they're activated by flu virus for 24 hours, they differentiate into 3 subsets that can be distinguished based on those surface markers. These 3 subsets, P1, P2, and P3, have distinct transcriptional signatures, and they have distinct functional profiles. The interferon alpha production is mostly in the P1 subset of the plasmacytoid dendritic cells. And so given that high levels of type 1 interferons are what we really want for cancer immunotherapy, we really want agents that will induce that P1 subset, or at least that's a good working hypothesis. So the reason I'm telling you all this is that these investigators also looked at the induction of these 3 subsets of PDCs with various agents, including R848, the gold standard TLR7/8 agonist, CpG-A, CpG-B, and CpG-C. And to make things easy for you in terms of distinguishing the percentage of PDCs that differentiate into the P1 subset with each of these different agents, I put this on a table so you can see that the CpG-A is the only agent that induces more than half of the PDCs to go into the P1 subset. Now, of course, what's really happening in tumors is going to be different, and it's not just this one subset. It may be a balance between different subsets that have different roles. Maybe the ratio of different subsets is more important. We really don't understand all of the details of this, but what we can say at this point is that the CpG-A has a unique biologic effect on PDC differentiation. One way to look at this is in vitro in human PBMCs. This is data from our collaborators at the University of Iowa, just taking PBMCs and looking at different innate immune activators, and then assaying the supernatants at 24 or 48 hours for interferon alpha. And you can see CpG-A induces by far the highest interferon. This is the CpG-B that we were developing back in the Pfizer days, a clinical CpG-C, and again, the gold standard TLR7/8 agonist R848. What the other agents induce more of are the inflammatory cytokines, IL-6, TNF, IL-1 beta, as well as IL-10. We think that the profile that you really want for cancer immunotherapy is focused on type 1 interferon. Now, the second key point about Vidutolimod is the delivery in a virus-like particle. This is a bacteriophage. It's not infectious for human cells, and it forms very nice icosahedral structures that are extremely stable once formed. They're small in size, so they traffic easily through interstitial fluid to draining lymph nodes, and they stimulate an antibody response after the first dose. In contrast to oncolytic viruses, where antibodies to the virus are neutralizing, antibodies to the bacteriophage are actually facilitating, and they provide an enhanced systemic effect that you can easily see in mouse models. If you implant tumors on both flanks of a mouse and inject one of the tumors with either naked CpGs or Vidutolimod, you get the same level of regression in the injected tumor. But I'm showing here uninjected tumors, where the VLP delivery provides an added systemic effect compared to the naked DNA. And this has now been published in 2 other mouse models. So this VLP delivery is giving an added effect. What we think is happening to mediate that is that in a tumor following injection, antibodies bind to the VLP once they've been induced. Those mediate uptake into the PDC through the stimulatory Fc receptor, Fc gamma R2A or CD32. And they then escort the CpG-A cargo into the early endosomal compartment where TLR9 is activated, converting the PDC into this interferon-secreting machine, and also recruiting and collaborating with other types of immune cells in the tumor, including conventional dendritic cells, which can cross-present antigens that are released from the PDC in exosomes, and also tumor-associated macrophages that can be activated by the interferon and potentially also cell-cell contact to secrete CXCL10. Together, these stimuli are what drive this very strong systemic T cell response. Now, those T cells, however, do have the PD-1 marker on their surface. Hassan Zarour and colleagues were the first to publish this. And so that's why we believe it's very important to develop the drug in combination. And let me just show you the monotherapy data from our trial in PD-1 refractory melanoma. These are the individual responders in that trial. And the median duration of response here is 5.6 months. So it's a limited duration, but certainly encouraging to see this level of monotherapy activity. But here's the combination swim plot. In this case, we have 19.9 months. So the way we think about this is that Vidutolimod is inducing the anti-tumor T cell response. PD-1 checkpoint inhibition is allowing those T cells to persist and to maintain responses for long periods of time. The systemic nature of those responses is shown, for example, in this patient, which was one of John Kirkwood's patients from Pittsburgh, a woman who had a single lesion injected in an inguinal lymph node, and then had very nice regression of large lung metastases that's been durable. And if you look across all of the responders in the trial who had non-injected target lesions, the mean regression in non-injected target lesions is very comparable to the mean regression of the injected lesions. So where do we go from here? This is the trial that I've just been telling you about. We've started phase 2 trials in combination with Nivo, both in PD-1 refractory melanoma and in first-line melanoma. And we're very much interested in having further discussions with investigators who might be interested in collaborating with us in these studies and also in the ongoing translational work to get a better understanding of how this drug is affecting the tumor microenvironment and how it can be applied more broadly to cancer immunotherapy. I put the contact information here for our Chief Medical Officer, Jim Wooldridge, Dmitry Bobolov, who's present at this meeting, and of course, please also feel free to reach out to me. Thank you for your interest.